Yesterday, I have completed some of the topics. Now, I am going to share this topic. Excuse me. Estimation of allele frequency dominant conformant cases, multiple alleles. The main objective is concept of dominance allele, multiple allele, estimation of allele frequencies. A dominant allele is variation of a gene that will produce a certain phenotype. Even in the presence of other alleles, a dominant allele typically encodes for a functioning protein. The allele is dominant because one copy of the allele produces enough enzyme to supply a cell with the allele. Plenty of a given product. Some rarely on a product being created, like pregnant molecules. Okay. Pigment molecules for hair color, ion channels for proper cell functions, and other traits that rely on a functioning thing everyone. Other traits rely on the lack of an enzyme or an enzyme that is not functioning effectively. The lack of the product of the enzyme creates an entirely different phenotype. So when a dominant allele is completely dominant over another allele, the other allele is known as recessive. However, that there are also situations of incomplete dominance and codominance. In incomplete dominance, two dominant alleles can mix to create a third phenotype, a sort of mix between two phenotypes. So think a white and red flower alleles mixing in a heterogenotype to produce a pink flower. In codominance, different alleles are expressed in different areas, creating unique pattern. Think of codominance as a spots on a curve. To describe an allele as a dominant allele, you must reference another allele. Dominant and recursive or just descriptions of a relationship between alleles. Some alleles of a gene complete differently with different alleles. They may be dominant to one allele and recursive to another. It all depends on the protein they create. How those proteins interact with each other and how the entire system interacts with the environment. While most simple examples given are of a purely dominant recursive relationships, in reality the interactions are much more complex. Complete dominance, incomplete dominance. In case of complete dominance, a dominant allele completely hides the effect of recursive allele. Homogeneous individuals. Homogeneous Homogeneous dominant individuals have two dominant alleles is present, and the phenotype only reflects the action of the recursive alleles. In the heterogeneous individual, the phenotype appears to be the same as in the homogeneous dominant individual. This is the defining feature of complete dominance. The dominant allele completely hides the presence of the recursive allele to observe. This can be seen in a pea plants. In the color of their peas, the dominant allele produces a yellow pigment to the enzyme it codes for. One allele is sufficient to produce enough yellow pigment to make the entire plant look yellow. The green allele produces a non-functional enzyme, and no yellow pigment is created in a homogeneous recursive plant. The yellow allele is dominant to the green allele, and the green allele recursive to the yellow allele. The picture below shows the cross between a heterogeneous individual on the left and a homogeneous recursive individual on the. So this is incomplete dominance picture. Okay. So multiple alleles. In the real world, genes often comes in many versions. Alleles are always fully dominant or recursive in one another, but may instead display codominance or incomplete dominance. According to Alter, there are more kinds of gene which occupy the same locus are referred to as multiple alleles. Alleles are alternative forms of a gene, and they are responsible for difference in phenotype expression of a given trait. Brown eyes versus green eyes. A gene for which at least two alleles exist is said to polymorphic. Instances in which a particular gene may exist in three or more allele forms are known as multiple allele conditions. It is important to note that while multiple alleles occur and are maintained within a population, and individual possess only two such alleles or equivalent see on homologous chromosomes. Estimation of allele frequencies and autosomal gene locus may have condominant alleles, dominant and recursive alleles, or multiple alleles. If one decides to estimate the gene frequencies for each of these kinds of autosomal alleles in a given population, he has to adopt the different methods. HW helps us to estimate the allele frequency by G counting method. How many alleles or genotype contains. So calculation of gene frequency is condominant alleles. When condominant alleles are present in two allele system, each genotype has a characteristic phenotype. The number of each allele is both homogeneous and heterogeneous. Conditions may be counted in a sample of individuals from the population and expressed as a percentage of total number of alleles in a sample. If the sample is representative of the entire population concentration, then we can obtain an estimate of the allele frequencies in the gene pool. In a given sample of n individuals in each D homogeneous for one allele A dash A or heterogeneous A square and or homogeneous plus array n equal to D plus H plus R. Since each of n individuals are diploid in this locus, the two n alleles represented in the sample. Each AA genotype has two A alleles. Heterogeneous have only A allele. Here, P represent the frequency of capital A allele and Q represent the frequency capital A2 allele. P equal to 2D plus H by 2N equal to D plus H by 2 by N. Q equal to of H plus R by N. The problem N blood type truly furnishes a useful example of a series of phenotype due to pair of condominant alleles. None of three possible phenotypes M, Mn, and N appears to have selection value. These frequencies of the two alleles for a sample from a group of Indians living in Telangana, Andhra Pradesh, Tamil Nadu, and Karnataka can be calculated by following ways. The sample of 6,129 people includes the following three groups according to phenotype and genotype MN system. To calculate frequencies of two condominant alleles L, M, L, N, it should be kept in mind that these 6,129 persons process total of 2n equal to 2 into 6,129 equal to 12,255 genes. The number of L alleles, for example, 2D plus H, 1787 plus 7087 plus 3039. Thus, calculation of the frequency of LM alleles is follows. LM 2D plus H by 2N equal to 0.5395. LN 0.4605 is Q. Thus, the frequency of two codominant alleles is in the sample almost equal. And this is reacted in the close opportunity of 1:2:1 ratio, which is a sample examined for which codominant allele lengths. So gene frequencies expressed as decimals may be used directly to state probabilities. Probability is a function that represents the likelihood of occurrence of any particular form of the event. If we can assume this sample to representative of the population, then there is a probability of 0.5 that of chromosomes bearing the pair of alleles, and anyone selected randomly will be gene L and 0.4605 that it will be L. Let P represent the genotype frequency of L, and Q represent the frequency of Ln, LA. Then the frequencies of the genotype to be expected to the population are as follows. Expansion of HW formula P plus Q whole square equal to P square plus Q square plus 2PQ obtained into LM and LM gene frequencies in a population. So genotype, phenotype, genotype frequencies. LMN, LN, LM, P square equal to 0.2. LMN, LNM, MN, 2PQ 0.49. LN, LN, N, Q square 0.2121. Next, statistical genetics. Approach to equilibrium for X-linked gene. Natural selection, mutation, and genetic drift. Equilibrium when both natural selection in mutation. So approach to equilibrium for X-linked gene. After studying this unit, the student should be able to know the importance of body well-being law. No approach to equilibrium for X-linked gene. According to the Horde, we bring the frequencies of genotype or equilibrium depend on the frequencies of alleles. When a population is wholly Weinberg equilibrium, the properties of genotype are determined by frequency of alleles. To calculate allele frequencies of a gene X-linked loci, we apply the same principles. However, we must remember that a female possesses two X chromosomes and therefore has two X-linked alleles, whereas a male has only a single X chromosome and has X-linked allele. Suppose there are two alleles, X-linked locus, XA and XS. Females may be their homogeneous XA, XS or X or heterogeneous, or males or um, homogeneous XA, XY or XY. To determine the frequency of the X allele, we must count the number of copies of the XA. We multiply the number of XA females by 2 and add the number of XA females and the number of XA males, when then divide the sum by the total number of alleles, the locus, which is twice the total number of females plus the number of males. P equal to, similarly, frequency Q equal to the frequencies of X-linked alleles can also be calculated from genotype frequencies by adding the frequencies of the females that are homogeneous for the allele of the frequencies of the females that are heterogeneous for the allele, and the frequency of males heterogeneous type allele. So where P is f of xa, Q is f of xa. If you remember the logic behind all of these calculations, you can determine allele frequencies for any set of genotype, and it will not necessarily to memorize the formula. So example problem, the human MN blood type antigenes are determined by two condominant allele LM LN, where MN blood types and corresponding genotype 398 from a certain population. So phenotype MM, MN, NN, genotype LM, LM, LN, 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 LN. So number is 182, 172, 144. Calculate the allele and genotype frequencies of the MN locus for a given data. Phenotype, genotype, number. The genotype frequencies for the population calculated with the following genotype frequency, where F is 0.4, 0.4, 0.11. The allele frequencies can be calculated from either the numbers of frequencies of the genotypes. To calculate allele frequencies from number of genotypes, we add the number of copies of the allele and divide the number of copies all alleles of the locus, where P equal to 0.6, Q equal to 0.3. To calculate the allele frequencies from genotype frequencies, we add the frequencies of the homogenotype for the genotype 2 of the frequency of each heterogeneous that contain the allele, where P equal to 0.6 and Q equal to 0.3. So how they Weinberg expectations for X-linked locus. For an X-linked locus with two alleles X and X, there are five possible genotypes XX, 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 XY. Female possess two X-linked alleles, and the expected proportions of the female genotype can be calculated by using the square of the allele frequencies. If the frequencies of XA and XS are P and Q respectively, then the equilibrium frequencies of the female genotype are P plus Q whole square equal to P square plus 2PQ plus Q square. Males have only single X-linked allele, and the frequencies of male genotype P, we notice that their respective frequencies are the proportions of the genotype, so males and the females, rather the proportions axing the population. The P square is the expected proportion from female with the genotype XX. Females make 50% of the population, than the expected proportion of the genotype in the entire population is 0.5 into P square. The frequency of X linked recursive trait among male is Q, where the frequency among females is Q square. When an X-linked allele is uncommon, the trait will therefore be much more frequent in males than females. So this is a next chapter is a simple one. Natural selection, mutation, and genetic drift. the fundamental model, fitness. So thank you.